TeamBST from Brainstorm Therapeutics is integrating advanced machine learning with patient-derived midbrain organoids to discover disease-modifying therapies for Parkinson's disease. By combining midbrain organoids with foundation model-based network analysis, we fine-tuned SCGPT on familial PD organoid data, creating a model that accurately reflects dopamine neuron degeneration. This enables detailed mapping of disease mechanisms and identification of new therapeutic targets and biomarkers often missed by traditional methods. Our CNS drug discovery platform at Brainstorm utilizes patient-derived brain organoids. These organoids recapitulate complex disease pathologies in a model that's amenable to high-throughput screening using clinically translatable biomarker endpoints. We complement this approach using advanced network medicine to discover novel druggable targets and biomarkers, and to stratify patient populations for clinical trials. For our hackathon project, we focused on Parkinson's disease, a progressive terminal neurodegenerative disorder characterized by the loss of midbrain dopamine neurons. PD affects 10 million people worldwide, making it the second most common neurodegenerative disorder. But despite substantial investments in research, there are no approved disease-modifying therapies due to the lack of predicted translational models for drug discovery. Our patient-derived midbrain organoids accurately simulate dopamine neuron degeneration, helping to fill this critical gap. Our goal for this hackathon was to build a foundation model-based organoid gene network to improve Parkinson's disease drug discovery. Midbrain organoids from patient-derived GBA1 mutant iPSCs exhibit robust and reproducible dopamine neuron loss. As shown in the left panel, we established the reproducibility of gene expression across organoids from different patients and different batches by analyzing bulk RNA-seq data. Traditional gene set enrichment analysis of this data gave a high-level overview of critical dysregulated processes. These results confirmed the ability of the midbrain organoid platform and the integrity of the data that we'll use to build our network. For this project, we developed a foundation model-based network analysis workflow, utilizing published pre-trained single-cell foundation models, including SCGPT and GeneFormer within the NVIDIA BioNeMo framework. By fine-tuning the model with midbrain organoid data, we created a network that captures Parkinson's disease-specific biological processes, enhancing target discovery and translational research. The workflow includes dimension reduction, community detection, and cluster annotation, allowing us to identify disease-specific gene clusters linked to known biological processes. The established network enables the exploration of gene expression studies, enhancing our understanding of the disease model and drug mechanisms. We also created an interactive website for analyzing the organoid network, allowing users to evaluate gene clusters and disease mechanisms. Different nodes represent genes, and they cluster by similar expression profiles. For example, red nodes here indicate upregulated genes in GBA1 PD organoids, while blue nodes show downregulated genes, reflecting changes consistent with dopamine neuron loss and neuronal precursor function. We validated our approach by using rigorous benchmarking to compare different foundation models, including SCGPT and GeneFormer, as well as traditional gene clustering algorithms, such as HCWGCNA, to assess their performance in capturing PD-relevant pathways. The fine-tuned SCGPT model outperformed all the others in detecting processes critical to PD pathology, such as neurogenesis and cell cycle dysregulation. Our patient-derived midbrain organoid models effectively captured major pathogenic pathways in PD, especially in dopamine neurons. Bulk RNA-seq analysis revealed consistent downregulation of genes involved in neurogenesis and cell differentiation, aligning with findings in actual PD patient tissue. We also discovered cell cycle dysfunction, likely resulting from disease-related defects in neuronal precursor differentiation. In the single-cell sequencing, we saw consistent changes in neurogenesis and cell differentiation in two different patient-derived organoid models, which carry distinct mutations in the GBA1 or LRRK2 genes. We also saw processes that differ between these two genotypes, such as lipid metabolism in the GBA1 organoid and oxidative phosphorylation in the LEC2 organoid. This highlights the platform's potential as a clinical trial in a dish for Parkinson's disease. In conclusion, our foundation model-based network analysis utilizing human iPSC-derived midbrain organoids has successfully captured key biological processes involved in PD progression. The SCGPT fine-tuned model offers new opportunities for drug target discovery and biomarker identification. Future steps include using this platform for in silico perturbation screening to simulate drug responses and identify potential therapeutic candidates and biomarkers. We plan to integrate models like NVIDIA BioNeMo version 2 and expand our analysis to other neuropsychiatric and neurodevelopmental disorders, transforming drug discovery in these fields. During the hackathon, we formed a collaborative team, including members from Brainstorm Therapeutics, alongside Birkin and Anna, who are experts in machine learning and data processing, and Jens, who provided us with valuable organoid data sets. Thank you for your attention, and feel free to reach out to us if you have any questions.